Hey everyone, this is my AP Biology Chapter 13 review, which is going to cover meiosis and the methods of genetic variation that happens during meiosis, so like independent assortments and the other two. So first, it's important to go over just generally what is meiosis and some miscellaneous definitions. So meiosis is the process by which chromosomes are going to be reduced into basically 4 genetically different daughter cells, which allows for sex cells to be produced for reproduction, so eggs and sperm. So the reason that this reduction is necessary through meiosis is if you have a diploid cell and you try to convert that diploid cell into a sex cell, that is likely going to lead to genetic abnormalities, genetic problems within the baby. So you need to have the haploids containing half the amount of chromosomes in order for reproduction and the future baby to, be free of problems, right? So again, haploid just means one set of chromosomes, so 23 in humans, and diploid, it means 2 sets of chromosomes, so 46 in humans. Gametes are the sex cells in males and females that allow for the passage of genes to the next generation. So the egg for the female, the sperm for the male. The somatic cells are basically all of the non-gamete cells, so all of the cells in your body pretty much, except for the egg and the sperm. And most, if not all, of these cells have 46 chromosomes in humans. And then you have the zygote, which is the cell resulting from the fusion of the egg and the sperm. Then you get to meiosis 1, which is where homologous chromosomes separate. So homologous chromosomes are pretty much just two chromosomes attached to each other. So this is pretty similar to the 4 steps of meiosis. So you have prophase, metaphase, anaphase, telophase, but this just happens twice is the main difference. So you have interphase. So that's just where the cell goes through G1 phase, so growth, checkpoints, DNA synthesis in the S phase, G2 growth, and then checkpoints. So then you get to the first main phase in meiosis 1, you have prophase 1, which is where the chromosomes align into homologous pairs. And this is where crossing over happens because the chromosomes that align into the homologous pairs are random. So this contributes to genetic variability. Metaphase 1, the nuclear membrane completely disappears and the spindle fibers form fully. Homologous chromosomes also attach to the spindle fibers. And this happens in the middle of the cell, obviously, as shown by this image down here where it says metaphase 1. Anaphase 1, the homologous chromosomes break away into the opposite sides of the cell. And then telophase 1, the cell splits, the nuclear membrane reforms, the spindle fibers are going to disappear. So that is clearly shown with this image right here. Then you have meiosis 2, which is where the sister chromatids are going to separate. So now you have one chromosome is going to separate into 2 halves of the chromosome, which is the sister chromatids. So this is the exact same process, really, again. So prophase 2, the spindle fibers form. Metaphase 2, the sister chromatids attach to the spindle fibers on the edge of the cell and migrates towards the middle. Then you have anaphase 2, which is where the sister chromatids break apart and migrates to opposite sides of the cell. So really the exact same thing. And then you have telophase 2, the cell splits, nuclear membrane reappears, and the spindle fibers reappear. And then finally, you have sources of variation in meiosis. So we talked about crossing over already. Happens in prophase 1, where the homologous chromosome, or the 2 chromosomes that randomly find the homologous chromosome pairs, that process is random. So that creates genetic variability. You have random assortment of chromosomes, which occurs during metaphase 2. So when the sister chromatids align at the spindle fibers in the center. So the pairs of these sister chromatids that line up to become just attached to each other, I guess. That process is fully random. It's incorrect on the slide, sorry about that. And you have fertilization, which is the process by which eggs are randomly fertilized by the sperm, but there's billions of sperm cells and only one of them is going to reach the egg. So that process is random. Basically, which sperm is going to reach the egg, that process is random. So all of these variation methods create an exponential number of possibilities for genetic variation. And that is the end of chapter 13. So I'll see you all in the next review video, which is chapter 14. The format for the next video is going to be a bit different. I'm going to do it on notability and just a voiceover, since it's going to be more of the genetic problems and just going through them. So it's much easier to do that in notability. So thanks for tuning in. I'll see you next time.